Ever get that feeling where you just want to like really get to the bottom of something?
Yeah.
Not just the surface facts, but the deeper meaning, like how it all fits together, you
Yeah,
know?
yeah. Totally.
That's what we're doing this in this deep dive, going deep into genetics.
Oh, wow. Wow.
A field that has just like completely changed the way we understand life itself.
It really has.
And it's also like a perfect area for the kind of questioning that philosophers love.
Oh, absolutely. I mean, Evelyn Fox Keller even called the 1900s the century of the gene.
Oh, century of the gene.
And when you think about all the philosophical questions that come up, it's clear why.
Yeah.
Genetics isn't just biology. It touches on like our whole idea of what inheritance is, you know,
Wow.
what identity means, even our place in nature.
It's so huge. And speaking of huge, have you seen how much philosophers have written about that?
Oh, I know.
There are over 100 entries in the Stanford Encyclopedia of Philosophy that mention genetics.
That's incredible.
It's mind-blowing. It just shows you how much this science has sparked philosophical thought.
It's amazing.
So for everyone listening today, our goal is to give you a good introduction to this whole area where genetics and philosophy meet.
Yeah, that's the
We
goal.
want to unpack the key ideas and really dig into the big questions that come up when you look at genes through a philosopher's eyes.
That's right.
It's like we're going to equip you to think about genetics like a philosopher.
Exactly.
We'll look at the history of genetics, then we'll dive into some of the really fundamental philosophical debates about the core ideas,
Yeah.
and finally, we'll look at the bigger implications of this whole field.
Okay, sounds good.
All right, so let's go back to the very beginning. Genetics as a distinct field really started taking shape around 1900, right?
Right around then, yeah.
It's kind of wild to think that Gregor Mendel, who did so much of the early work, never even used the words gene or genetic.
I know, right?
Back in the 1860s, he was doing all these careful experiments with pea plants. And that's how he discovered those basic patterns of inheritance.
Yeah, the fundamentals.
Like segregation, where offspring get one copy of each trait from each parent.
Right.
Independent assortment, where those traits are inherited independently of each other.
That's another big one.
And dominance, where one version of a trait can mask another.
Oh, yeah, like the classic dominant and recessive traits.
Exactly. Now, philosophically, this was a huge shift because before, people thought inheritance was like a blending process.
Yeah, like missing paint or something.
But Mendel showed it was based on these separate units, which made it possible to use math to model it.
That's a really important point. But it's funny how his work went unnoticed for a while.
Yeah, it's true. It wasn't until the turn of the century that other botanists rediscovered his findings, and then suddenly these discrete hereditary factors became the big thing.
Everyone was talking about them.
That's where William Bateson enters the scene.
Bateson.
He really saw how important Mendel's work was for evolution, and he came up with the terms genetics for the field and gene for that heritable factor.
The name stuck.
They really did. And he and his team were crucial in showing that Mendel's principles weren't just for peas. They applied way more broadly.
Much broader, yeah.
So you have genetics emerging, and it leads to these two main lines of investigation.
Okay, two paths.
One was focused on the physical basis of these genes, like what are they made of?
Yeah.
Where are they located?
Uh-huh.
How do they actually work to create these patterns of inheritance that we see?
Those are the big questions.
Thomas Hunt Morgan's work with fruit flies at Columbia was key for this.
Fruit flies.
He provided strong evidence that genes are actually on chromosomes. And his team even figured out how to map their relative positions.
Wow, mapping genes on chromosomes, that's
It
amazing.
was revolutionary. By breeding tons of fruit flies and even causing mutations, they were able to pinpoint the chromosome as the location of genes. But then there was this other massive question that kept coming up. How did this discrete inheritance that Mendel described fit with Darwin's idea of gradual evolution?
Yeah, that's a big one. The two theories seem kind of at odds.
Right. Darwin envisioned slow, continuous change building up over time,
Gradual change.
while Mendel's factors seem more like on-off switches.
It was a puzzle.
It took brilliant people like R.A. Fisher to bridge this gap. Starting around 1918, he showed mathematically that if you consider many Mendelian factors influencing a single trait, the natural selection acting on these little variations could actually lead to the gradual changes Darwin talked about.
So that's how they reconciled those ideas.
It's a crucial insight from a philosophical perspective
Totally.
because it connects these seemingly separate views of Darwinian gradualism and Mendelian inheritance, creating a more unified and measurable way to understand how life changes over time.
A unified theory.
And that's how the modern synthesis was
For the
born.
modern synthesis.
This powerful framework that sees evolution as changes in gene frequencies within populations over time.
Okay, I'm following.
Evolutionary biology became much more quantitative, relying on mathematical models to explore the impact of things like migration, genetic drift, and natural selection.
It's not just describing what's similar between organisms anymore.
Exactly. What's so fascinating philosophically is how trying to understand the basic mechanisms of inheritance and evolution not only established genetics as a field, but also completely reshaped how we understand the process of life diversifying.
It's like changing the foundation of our understanding.
Now let's jump ahead a bit.
Okay, fast forward.
We come across some fundamental philosophical challenges, starting with a seemingly simple question. What exactly is a gene?
Yeah, what is a gene? Seems straightforward, but
It turns out it's way more complicated than that early idea of one gene neatly determining one trait.
really gets complicated.
As molecular biology advanced, especially after we discovered DNA's double helix, the simple gene concept started breaking down.
Things got messy.
Scientists found things like overlapping genes, where the same stretch of DNA could code for different proteins depending on how you read it.
Oh, wow. Overlapping genes.
Then there were split genes, where the protein coding regions called exons are interrupted by these non-coding sequences called introns.
Exons and introns.
And to make things even more complex, there's alternative splicing, where those exons can be combined in different ways to make multiple proteins from the same gene.
Like a choose-your-own-adventure book for proteins.
Kind of. It's like a recipe book, where some paragraphs, the exons have the key instruction,
Okay.
and others, the introns, are like optional notes that you can include or not.
I see, I see.
Alternative splicing is like choosing different combinations of those instruction paragraphs to make slightly different versions of the final dish, which is the protein.
That's a really cool way to think about it.
It really messes up that clean one-to-one relationship between a gene and a protein, right?
Yeah, it does.
It shows just how complex things are at the molecular level.
So much complexity.
And this is where philosophers like Lenny Moss stepped in to make crucial distinctions, like between gene P and gene D.
Gene P and gene D. Can you explain that?
Sure. So Moss argued that we're actually using two different concepts of the gene.
Two different concepts.
One he calls gene P, which stands for instrumental preformationism.
Instrumental preformationism.
It's basically defined by its link to a specific trait.
Yeah.
You know, like when people say the gene for cystic fibrosis.
Oh, I see.
In this sense, the gene is mainly a tool for predicting if someone will inherit that trait or disease, often without fully understanding how it works at the molecular level.
So it's more about tracking the patterns of inheritance over generations.
Exactly. Then there's gene G, the developmental resource.
Developmental resource.
This refers to the gene as a specific DNA sequence.
So like the actual physical gene.
Right. For cystic fibrosis, gene D would be that particular DNA sequence that's mutated,
Okay.
but it acknowledges that this gene doesn't act alone. It's just one factor among many that contribute to the disease. So it's not the only thing causing it.
Not at all. This distinction is important philosophically because it highlights the different ways we think about genes, either as tools for predicting traits or as actual physical entities raising questions about how we explain things in biology.
So gene P is about observable inheritance and predictions, while gene D is grounded in the actual molecular machinery.
Right.
But then you have philosophers trying to bring these perspectives together, like Eva Niemann held with her process molecular gene concept.
Neumann held. Interesting.
She wanted to unify these different ideas by defining a gene as the process that leads to making a specific protein.
The process, not just the thing.
So for cystic fibrosis, the gene would be the whole process, not just the DNA, but all the molecules and steps involved in turning that DNA into the protein that's supposed to be an ion channel.
Okay, whole shebang.
And the disease happens when a specific DNA sequence messes up this process.
So it's a breakdown in the process.
It's a much more dynamic view, seeing the gene as something active rather than static. This whole debate shows how our understanding of even something basic like a gene is always being shaped by new scientific findings, and how philosophy helps us make sense of those concepts.
It's like philosophy is helping to refine our scientific understanding.
Absolutely. And this careful philosophical thinking becomes even more crucial when we consider what we expect genes to be able to do.
What we expect them to do,
You
yeah.
remember all the excitement surrounding the Human Genome Project?
Oh, yeah, the Human Genome Project.
People were so hopeful that we'd quickly find genes for all sorts of diseases. and that genetic cures were right around the corner.
There was so much hope.
The media was buzzing with talk about deciphering the book of life and how personalized medicine was just a matter of time.
It felt like a new era.
It felt like we were on the verge of unlocking everything through genetics.
But it turned out to be more complicated than that.
So much more complicated.
Yeah. When scientists started doing these huge studies called genome-wide association studies, the picture became much more complex.
What did they find?
Well, for most common diseases like heart disease or diabetes, there wasn't just one gene causing it, or even a handful of major ones.
Not one single gene.
Nope. They found many different spots in the genome that all play a tiny role in increasing the risk.
So it's not as simple as finding the bad gene.
Not at all. And even for single gene disorders like cystic fibrosis, finding a cure has been incredibly hard, even though we've known the gene for decades.
It's surprising.
Yeah, it highlights the limits of just knowing the gene.
So this difference between the initial hype and the more complicated reality led to some really important philosophical critiques, especially around the idea of genetic essentialism.
Genetic essentialism.
This idea that our genes are the main thing, or maybe even the only thing, that determines our traits and our future.
Like our destiny is written in our genes.
Exactly. Critics argued that oversimplified language like saying the gene for X could be misleading and even harmful because it overemphasizes genetic explanations and solutions while ignoring environmental, social, and lifestyle factors.
So it's not all in our genes.
Right. And this focus on genes might have even directed research funding away from other areas of medicine that could have helped people more quickly.
Yeah, that's a good point.
We also can't forget about the influence of metaphors.
Metaphor.
Yeah, like the idea of the genome as a program or a blueprint.
I've heard those before.
They might be helpful at first for grasping the concept, but they come with a lot of baggage. Like they imply a level of predetermination and a linear flow of information that doesn't really match how dynamic and interactive biological development actually is.
So those metaphors are oversimplifications.
They suggest that all the information needed to build and run an organism is neatly packaged in the DNA sequence, which philosophers and biologists like Paul Griffiths and Eva Jablonka have argued against.
Okay, so it's not that straightforward.
They say biological information is spread out over many levels, including things like epigenetic modifications, cellular structures, and even the environment.
So many levels.
Now, C. Kenneth Waters provides another philosophical perspective focusing on the concept of causation in genetics.
Causation, interesting.
He distinguishes between potential difference makers and actual difference makers in the context of controlled experiments.
Okay, break that down for me.
In a well-designed experiment, many things could potentially influence a trait.
Lots of possibilities.
But it's the specific genetic change that actually causes the difference between the experimental group and the control group.
So you isolate the one thing that made the difference.
Exactly. Waters says this helps explain why genetics has been so powerful in the life sciences.
Because you can isolate the cause.
It lets you pinpoint and manipulate specific causal factors in a very controlled way.
That makes sense.
But the philosophical counterpoint is that genes aren't always the only actual difference makers, even in those tightly controlled experiments.
There are still other factors.
The environment, even subtle differences in development can play a role. It's a reminder that even when we're focusing on genes as causes, they're always operating within a larger context.
Always part of a bigger system.
And this more nuanced understanding is reflected in how different scientific fields study genes today.
How so?
In fields like psychiatry, conservation biology, even research on coral reefs, genes are seen as one important level of analysis among many.
Not the only level.
It's a more integrated and less gene-centric view than we had in the early days of molecular biology.
So the gene isn't this all-powerful controller, it's one piece of a big puzzle.
Exactly. Now let's shift gears a little and think about the philosophical implications of natural selection when we view it through this genetic lens. Darwin initially described natural selection as acting on individual organisms, right?
Survival of the fittest individual.
That was the basic idea. But then we see things like altruism, which seem paradoxical.
Altruism, like selfless behavior.
Yeah. Why would an organism act in a way that benefits others, potentially at a cost to its own survival or reproduction?
It seems counterintuitive.
It seems to contradict the idea of individual selection.
Right. Why would you sacrifice yourself for others?
The classic example is an animal giving a warning call to alert others to a predator, even though it might attract the predator's attention to itself.
putting itself at risk to save others.
It doesn't seem to directly benefit the caller.
That's the puzzle.
W.B. Hamilton came up with the idea of kin selection to explain this.
In selection.
He said if the altruistic individual is related to the ones that helps young, they share some of the same genes.
Because they're family.
So even if the altruist dies, its genes, including the ones that might make it altruistic, can still be passed on through its relatives.
So the genes survive even if the individual doesn't.
And this idea was further developed by people like G.C. Williams and Richard Dawkins with their gene's eye view of evolution.
Gene's eye view, I've heard of that.
This shifts the focus from the individual organism being selected to the gene itself.
The gene as the unit of selection.
Dawkins called genes replicators, the things that get copied and passed on through generations in individual organisms, just vehicles that carry those genes.
So we're just vessels for our genes.
From this view, the ground squirrel's alarm call can be seen as a strategy that benefits the genes for altruism, because those genes are likely in its relatives who are now more likely to survive.
So it's not about the individual squirrel, it's about the survival of those altruistic genes.
This gene-centric view really sparked debate about the levels of selection.
Levels of selection.
Is natural selection acting on genes, individual organisms, groups of organisms, or even whole species?
It's a complex question.
Some argued for group selection, saying groups with more cooperative individuals might outcompete more selfish groups.
Cooperation wins.
Others, like Stephen Jay Gould, thought selection could happen at the species level.
So many possibilities.
And then you have ideas like developmental systems theory, which challenges the whole replicator vehicle distinction and emphasizes the entire developmental system, including both genes and environment, as the basic unit of evolution.
It's not just nature versus nurture, it's both working together.
So the question of what's really being selected in evolution is still a hot topic for philosophers. Now let's move into the more applied and ethical realm of medical genetics. We briefly talked about the dark history of eugenics,
Eugenics, a very disturbing chapter.
which was based on a completely flawed and often prejudiced view of genetics.
A dangerous misconception.
It was rooted in deeply discriminatory beliefs about which lives were valuable, leading to horrible policies like forced sterilization and racist immigration laws.
Trying to control who reproduces based on flawed ideas.
When medical genetics and genetic counseling emerged in the mid-20th century, there was a deliberate effort to distance themselves from these dangerous ideas by focusing on individual choice and family autonomy in making reproductive decisions.
Empowering individuals and families.
Technologies like amniocentesis carrier screening and pre-implantation genetic diagnosis gave parents more information about their potential children's genetics, with the goal of helping them make informed choices about family planning.
More information to make decisions with.
And non-directiveness became a key principle in genetic counseling,
Non-directiveness.
meaning providing information without explicitly telling someone what to do.
Just presenting the options.
But scholars from disability studies have raised questions about whether this really broke free from eugenic thinking.
Interesting, how so?
They argue that simply offering screening for some conditions like Down syndrome or cystic fibrosis, but not for others, can subtly imply which conditions are undesirable and should be avoided.
So the choices presented aren't neutral.
And the ideal of being non-directive in genetic counseling is also tricky. Can it truly be value neutral when the very availability of these tests and how the information is presented can influence decisions?
It's hard to be completely neutral.
There's a worry that it could unintentionally reinforce the idea that disability is something to prevent at all costs.
Trying to eliminate disability rather than accepting it.
Critics point out that this focus on genetic screening can lead to the medicalization of pregnancy and birth, changing how parents view and prepare for their child.
It frames everything in medical terms.
There are also concerns about making existing health disparities worse if access to and use of these technologies differ between socioeconomic or racial groups.
Equity of access is crucial.
And of course, the growing amount of sensitive genetic data being collected raises huge questions about privacy and potential misuse.
Who has access to that data and how will it be used?
It's a complex ethical landscape where technologies designed to inform and empower people can also raise concerns about societal biases and fairness.
So many ethical considerations.
Now let's look at an even more cutting-edge area, human enhancement with genetic technology. We already make a distinction between treating disease and enhancing normal human capabilities.
Treatment versus enhancement.
That distinction gets really blurry and ethically challenging with genetic interventions.
Especially with things like CRISPR.
Especially with CRISPR-Cas9, this precise gene editing tool that makes it much more possible to intentionally change the human genome.
It's a powerful tool with huge implications.
The philosophical debate about the ethics of genetic enhancement has become even more urgent.
The hot topic.
Philosophers like Michael Sandel have voiced strong concerns, saying that this desire for genetic enhancement often comes from a kind of hubris, a dissatisfaction with the natural lottery of life.
Like wanting to control everything.
He values accepting our natural abilities and worries about what might happen to society if we try to engineer ourselves.
It could lead to all sorts of problems.
Jürgen Habermas has raised profound concerns about parents using genetic enhancement.
What are his concerns?
He argues that it could violate the future autonomy of the child by deciding their traits for them and limiting their freedom to shape their own life.
So it's about the child's right to choose who they are.
And of course, there's the dystopian vision of a future with genetic haves and have-nots, like in the movie Gattaca.
Oh, yeah, that's a scary thought.
It raises serious questions about social justice and equality.
A genetically stratified society.
But supporters of genetic enhancement like Julian Savulescu have a counter-argument.
What's their perspective?
He uses the idea of procreative beneficence,
Procreative beneficence.
suggesting that parents have a moral duty to use available technologies, including genetic ones, to try to give their children the best possible start in life.
To help them thrive.
He compares it to other things parents do, like providing a good education or a nurturing environment.
So it's like another way to help your child.
But critics still worry about who gets to decide what a better life is and see hints of eugenics in this pursuit of genetic perfection.
Yeah, it's a slippery slope.
The line between therapy and enhancement can get very blurry, and there's always the risk of unintended consequences for society.
That's a lot to think about.
It forces us to really examine our values and what it means to be human and what kind of future we want to create.
Big questions about humanity.
Lastly, let's talk about the complicated and sensitive relationship between race and genetics.
Okay, race and genetics.
We know eugenicists had a very flawed and racist understanding of race as fixed biological categories.
Well, a very dangerous idea.
Their belief in a hierarchy of races with inherent differences in intelligence and behavior has been completely debunked by science.
Completely discredited.
But there's still a lot of interest in understanding the genetic basis of human diversity.
Human diversity is fascinating.
Studies of human evolution have shown how different populations adapted to different environments, which led to variations in certain genes related to things like skin color or susceptibility to specific diseases.
So our genes reflect our evolutionary history.
This has led to the idea that racial categories might reflect groups with different genetic profiles linked to their ancestry and evolution.
So race is a reflection of genetic ancestry.
For instance, someone with African ancestry might have different genetic predispositions related to melanin production compared to someone with European ancestry, and also potentially different chances of carrying certain disease-related genes.
It's about understanding patterns in populations.
While proponents of this view stress that they're not trying to bring back racial hierarchies but just tracing different population histories and patterns of genetic variation, philosophers have raised some important challenges.
What kind of challenges?
For one, the genetic groupings from evolutionary studies don't always perfectly match our socially defined categories of race.
The lines aren't so clear.
And critics like Udell and Dorothy Roberts argue convincingly that racial categories are deeply rooted in social, political, and economic histories, not just biology.
It's not just about genes.
There's growing agreement among many philosophers and scientists that we need to separate race as a social construct from ancestry as a more biologically meaningful concept, especially in things like genetic testing and research.
That's a key distinction.
It highlights how even seemingly biological ideas like race are shaped by both science and social realities. It reminds us that we constantly need critical philosophical thinking to make sense of scientific findings.
It's an ongoing process.
As we wrap up this deep dive, what becomes clear is how deeply genetics is connected to fundamental philosophical questions.
It touches on so many things.
It challenges how we understand core concepts like the gene itself, the nature of cause and effect in biology, the forces driving evolution, and the very basis of a just society.
Genetics raises big questions.
From the birth of the field to the latest breakthroughs in gene editing, genetics keeps pushing the boundaries of science and raising huge ethical and societal questions. I hope this deep dive has given you a good introduction to this rich, ongoing philosophical discussion.
It's been a fascinating exploration.
As we move deeper into this second century of the gene, with our increasing ability to understand and manipulate the building blocks of life, it's clear that philosophy and genetics are intertwined.
They need each other.
It's not just an academic exercise. It's crucial for figuring out how to navigate the future responsibly.
Responsibility is key.
So for you, our listener, the takeaway is this. In wrestling with the implications of genetics, we're all, in a way, acting as philosophers.
We're all thinking about these big questions.
By questioning things, considering different viewpoints, and examining our values, we're actively shaping how we understand and use this powerful science.
We're shaping the future of genetics.
As we continue to unravel the mysteries of life, our collective responsibility is to use this knowledge wisely, with careful thought about its impact on everyone.
It's up to us to use this knowledge for good.
We have to be mindful of the consequences.
Absolutely.
It's a future we're all creating together.
And it's one we need to shape carefully.
Thanks for joining us on this deep dive.
It's been a pleasure.
We'll see you next time.
Until then, keep those philosophical gears turning.
And remember, genetics is more than just science. It's a reflection of who we are and who we choose to be.
A reflection of humanity.